Really said, hopefully.
All right, good morning, everybody. Welcome to another High Rounds. I hope everyone had a great July off.
Woohoo!
So before we get started, I was asked to make a quick announcement about San Diego CFAR Research Day. So they have a call for art submissions. The deadline is this coming Monday. And so they invite community members to share their stories through art, reflecting on how HIV has shaped their lives and identities. 1 to 3 selected pieces will be displayed in the Great Hall on the UC San Diego campus in La Jolla, where artists will have the opportunity to present the stories behind their work and their personal experiences with HIV and survivorship. Additional submissions not selected for display may be featured on our website. See submission guidelines and details in the attached flyer. So I can put the website in the chat later.
Okay,
Thank you.
great. Thank you. Thanks everyone for being here. Welcome to a new season of UCSD High Rounds. So first of all, I wanted to thank Jill Blumenthal for all of her work over the many years leading it. She has stepped down temporarily, but because I'm gonna make her work a little bit later this hour because I'm gonna have to leave a little bit early. But I will put out a plug that we are looking for someone to help perhaps lead High Rounds, or if you're interested in helping out, we're more than happy to use your assistance, first of all. And second of all, I think we are also looking, if anyone has talks that they're interested in doing, please reach out to us. Reach out to Marvin. You can send him an email. We are looking for someone for next month, at least. I think 1 or 2 talks if people are interested. So I have the pleasure of introducing Dr. Sara Gianella Weibel, who I think many of us know here. She's a professor of medicine and infectious diseases here at UCSD, where she leads a translational virology laboratory focused on HIV persistence and cure. She is perhaps best known for her leadership of The Last Gift, a unique rapid autopsy research program that studies HIV reservoirs throughout the human body. Today, she will discuss her work on sex and gender differences in HIV persistence and what these differences may mean for HIV cure strategies. So go ahead and take it away.
And then congratulations to Sara as of tomorrow, her half FTE, well-deserved start.
Thank you, Tim. I
Yes.
mean, this couldn't happen without your support. So thank you. Okay, so I'm really excited to talk about sex and gender difference in HIV, which I was just discussing with Marvin. I haven't done this talk in quite a while, and so I spent some time updating the slides, and really a lot has happened. So a lot of exciting stuff are happening. And I also wanted to kind of brag a little bit about the art piece that my daughter Emma made last year. And it is part of a publication, and it's called Love and Liberation, and it's in my office. Okay, so what we will discuss today is why have women been underrepresented in HIV research, but really research in general. Do sex and gender change HIV pathogenesis and persistence? And what does it mean for HIV cure? Okay, a little bit, just a brief history of inclusion-exclusion of women in research in general. So early, like in the 60s and 70s, women in reproductive age were routinely excluded from all kind of research because of concern around reproductive toxicity, variability in the menstrual cycle, concern about women's autonomy, and samples, and with the conclusion that sample size of study might be too large if we had to include women. And the FDA in 1977, they even mandated that excluding women with childbearing potential from participating in phase 1 and 2 clinical trial until reproductive toxicity study were conducted, basically to protect the women and their unborn fetuses. And so the vast majority of research were conducted in males and then extrapolated to women. And one of the main study that kind of really triggered this worry, and I don't know how much of you remember like the thalidomide, which right now is a drug that we use to cure many blood cancer. When it started, it was put on the market as a sleeping pill, and it was prescribed routinely to women. And so there is a whole generation of kids that were born with missing limbs. And this was big in Europe because I still remember the generation of my moms. There was occasionally someone that had a missing piece of an arm, and so because of that phenomenon. And I think this just really raised a lot of concern, and then these restrictions. And a lot of advocacy happened even in HIV, but not only, until early in the 90s. There was a committee who, of ethicists and advocates, they started to raise issues related to inclusion of women until in 1993. There is finally the NIH Revitalization Act that now determines that women and minority must be included in clinical studies. And this is in theory, we know that this doesn't happen as much, but at least that was the legal basis that right now there's no excuse to not enroll women. Going back to HIV, so even if the epidemics here in Southern California is driven by mostly men, in reality worldwide, more than 50% of the people with HIV are women, especially in Sub-Saharan Africa, where young women are disproportionately affected by HIV. And despite this, the vast majority of HIV cure trial in particular, because they're very sophisticated and expensive and require all sorts of infrastructure, they mostly take place in country, in the US or Western Europe, where there are not so many women. And so in HIV cure in particular, there is like a big underrepresentation of women. I would say probably in average around 10 to 11% of participants in cure trial across, and we will see some data later, are women compared to over 50% of the general population. We also know that the burden of HIV among women is highly unequal. And this is like some data, and these are data from the US, where we see that the vast majority of women living with HIV are really Black women than Hispanic and white. So there is also a big inequality of distribution of HIV. So regarding HIV pathogenesis, like there are many reasons to think that sex and gender will affect in some way. Like from the early of the epidemics, many studies have shown that women, for example, have lower viral load compared to men during acute infections and higher CD4 T cells. This is actually a study from the 80s, but many other studies have shown the same. But despite this, women progress to AIDS at a similar rate than men. And this was particularly important to know early in the epidemic when treatment eligibility for antiretroviral therapy was determined by viral load. This means that based on viral load, like only about 37% of women would qualify for antiretroviral therapy compared to 3/4 of the men. But then looking at the disease progression, women in proportion were doing worse after adjusting for the viral load. So this was already kind of known early in the epidemic. And why is this, right? There are many reasons. And obviously there are many socioeconomic, cultural reasons, like socioeconomic disparity, especially outside the US, the late recognition, like women, like usually don't pay so much attention to symptoms. They might delay going to the doctor. They have many competing priorities, antiretroviral pharmacokinetic differences. And this is one more place where a lot of our antiretroviral are studied in men and then applied to women, even if we know that women have different body composition. And so there are data showing that women tend to have a little bit more adverse event, and they do discontinue antiretroviral therapy more often. And also just a general barrier to adherence, attention, going to the doctors, and everyone. So we're all familiar with these barriers that a lot of women encounter. But so there are also very well-known biological mechanisms, and one of them is the double-edged sword of the interferon response. So interferon is one of our most potent antiviral responses. And what makes this interesting is that one of the genes that encodes this response is on the X chromosome. And in women, it's only partially inactivated. I don't know if you're familiar that because women have two X chromosomes, sometimes we have two genes and men only have one, but the second gene is supposed to get inactivated, but sometimes it doesn't get completely inactivated. And this is one of them. And basically, because of this incomplete inactivation, there is like a higher expression of interferon, which is good in acute infection because more interferon means we control the virus better, but over time, it's deleterious and it increases the risk of persistent inflammation and adverse events and mortality and morbidity. And so this is kind of the double-edged sword. And women are also known, even women without HIV, we have stronger response to vaccines and generally stronger antiviral responses to many viruses. And I know that this is a very busy picture, but I wanted to put it here to do a plug on Liz's recent lifetime review, which was such an art of piece of work. It's this. And it's a very long, very detailed review about the landscape of HIV reservoir in women across the whole life cycle from babies to menopausal transition. And we put together, I think, everyone that I knew in the field that was doing this type of research. And it's like it spans between virology, but also community and ethics. And it's a incredibly throughout complete piece of work. So if you are ever interested, you need to read this to know more. And to visit the most amazing figures, this being one of them. Basically, here the take-home message is that estrogen doesn't have only one effect on the immune system, but it's really highly dose-dependent. So low estrogen levels are more like the whole immune milieu. And this is like a simplified way to do it, because if you are a immunologist, it would probably kill me because like there are so many different nuances, but basically low estrogen, inflammation, pathogen clearance, high estrogen, immune tolerance, and tissue protection. And if you think about it, it makes sense because during pregnancy, like when you have very high estrogen is when you want to protect your baby and your kids. And so you don't want the immune system to attack your the foreign body. Okay, so now let's talk about sex and hormones and HIV reservoir. So based on everything we learned, like it's very likely that sex and sex hormone are likely to affect HIV persistence, right? Like what about the size of the reservoir, the activity, the reservoir composition? And so right now I will show you some data about this, and then I will dip a little bit more into reproductive aging and menopausal transition, because this is a topic that I'm particularly interested in for personal reasons, not only, but I also have another one to study exactly that transition, which was totally understudied, and recently a lot of people have turned their attention in the menopause, which is amazing, and especially now that we have an aging population of people living with HIV, there is a lot of awareness about the menopausal transition. Okay, so base sex difference in reservoir size. So like these are some, not all of the study that have looked at this, most of them cross-sectionally. So the evidence is mixed. Like some studies show a lower reservoir, some didn't see any difference. So I will say mixed evidence, maybe lower, but we're not sure. when we look at reservoir activity in terms of cellular HIV transcription or inducible virus, there is much more consistency. And almost, with maybe one exception, almost every study show a decrease in HIV transcription in women compared to men. And why is that? So this is like a really seminal paper for those of us that study HIV persistence from John Carnes, which is co-PI on my menopause arrow one. So what he did did an unbiased genome-wide screen because he wanted to see what compound might be suppressing HIV RNA transcription. And it was unbiased. So he did not in any way think estrogen. He wanted to just see what is out there. And out of hundreds of compounds, estrogen seemed to be one of the strongest inhibitor of HIV transcription. And so in this paper, he showed, and these are cell lines. First, he showed that when he tries to stimulate HIV transcription, and then he adds estrogen, and these are the two boxes that I highlighted here, like there is a huge decrease in HIV transcription after adding estrogen. But what makes it even more interesting is that this effect is sex-specific. So when he took cells from women or men, and he repeated the experiment, like on the left, we have the women, on the right, we have the men, you can see that when he tries to induce HIV transcription, and then he added estrogen, there was a almost complete inhibition of HIV transcription in women, and only about half of the cells were inhibited in men. So you can imagine how this could potentially have some effect when we try to do kick and kill strategies, which I think everyone is familiar with. One of the main way that people are trying to cure HIV is through trying to induce transcription and then kill the cells. So what, like, as a follow-up of this paper and this evidence, Dr. Eileen Scully designed the MOXIE trial, where she basically was using tamoxifen as a estrogen antagonist to see if, when you do kick and kill strategies in women, with or without tamoxifen, if you will be able to increase the kick when you block the estrogen receptor. Tamoxifen, most people are probably familiar. We use it in breast cancer treatment when they're estrogen sensitive to block the receptor. But I like this trial because it has multiple lessons learned. And lesson learned number one is that despite the fact that everyone was raising concern about not being able to enroll, she enrolled in a heartbeat, like all of the women in the US. And the second lesson learned is that SASC, which is the ACTG people that make this decision, they were very worried about reproductive toxicity. And so they basically forced Eileen to only enroll postmenopausal women. And so the trial ended up being negative. And she didn't see an effect. And what we hypothesize is that probably she just studied the wrong population because postmenopausal women probably lost part of the susceptibility. And so the whole trial didn't really answer the question it was designed to answer. But it was still a great trial. And we learned a lot of stuff, but it didn't really answer the question that it was designed. Okay, any questions so far? I will be talking about the menopausal transition. So the menopausal transition is actually, and like if we are interested in the effect of estrogen on HIV reservoir, like the menopausal transition is a really perfect life case because as you can see, like there is like a steep decline in estrogen starting around age 45 until women hit menopause around 51. So that window, like if we are able to collect longitudinal samples, we can really look at what happens within each woman in terms of immune response and HIV persistence. And so this was the first paper that we published, and I honestly think it was the seminal paper raising a lot of the current interest in menopause, because when we published this paper, nobody else was yet looking at menopause, and then a lot of people started looking at it. But here we had two cohorts, like we had one cohort of ACTG women and men, like 59 women, 31 men. And we looked longitudinally at HIV persistence, doing HIV DNA and cell-associated RNA. So it was a large-ish cohort with 80 people, but in the ACTG, they have not been consistently collecting all of the menopausal data. Now they do, but back in the day they didn't. And so what we did for the ACTG cohort, we based on age. So we just started at 45 and then collected sample over 10 years. And we assumed most of them had a decline in reservoir, in estrogen. But then we had 15 women that were very carefully characterized longitudinally, pre, peri, and post-menopausal, using stroke criteria and hormone, and we looked at the reservoir change. And as expected, I will say, what we saw is in the bigger ACTG cohort, we saw that there was a difference in HIV DNA dynamics over time, where the men were consistently declining and the women were having a plateau over time, and it was statistically significant. But then I think that what was really exciting, and I've seen this figure used in different talks, is that in the 15 women from the WISE cohort, we can see that inducible HIV RNA was increasing significantly as they went from pre to peri to post menopause. So if you think about the famous John Carr and Das paper, where estrogen inhibits HIV transcription, this makes a lot of sense. And so this means that women, as they age and they go through menopause, there is definitely more virus transcribing and bubbling and inducible virus going on. And what we're doing right now as part of the R01, and these are unpublished data, is we're looking, for example, is there clonal expansion of the HIV reservoir over time? And here, these are 6 donors. We have done more in the meantime, where you can really see that as women age, there is more clonal expansion of HIV DNA. This means that the virus is expanding, which makes sense, right? Because there is more transcription, and so cells get activated and they tend to expand and making the reservoir larger over time.
And how does this interact with CMV? Does that?
We haven't looked at CMV much yet, but I can imagine that CMV could play a role here, right? Because like over time, you might also lose a little bit control of CMV replication, and so CMV can also contribute to clonal expansion. I think that's a good question. This is a paper from Matthias Lichterfeld and Shu Yu. They have a lab at Harvard, and they do very sophisticated sequencing work. So what they tried to answer different questions, like they looked at not only quantity of the HIV reservoir, but quality and how does the reservoir shapes. And it was published here, in Science Translational Medicine, but they did presented this at CROI as an oral presentation a few years ago, and there was a lot of discussion around it. So here the study design was, they had 30 women and 35 men, and they followed them over a long period of time, and they do very sophisticated sequencing of the virus and integration site and immune profiling to try to compare the HIV reservoir landscape between men and women who stayed on suppressive antiretroviral therapy for a very long time. So they didn't find a big difference in the size of the reservoir. They, similar to the data that I showed us, they definitely show more clonal expanded virus in women. And they also, showed that there is lower phylogenetic complexity in women, which kind of plays together with the more clonal expansion, like there's less diversity. But what the most interesting to me is the landscape was different. And basically the virus was enriched in genes that were not transcriptionally active. We call them also epigenetic graveyards. Like the human genome, there are some genes that are active and some that are not active. And so it looks like HIV likes to hide in this graveyard, because if the gene is not active, HIV can hide better. So that's kind of a hiding strategy for HIV. And it looks like over time in women, this happens more often, which probably can make it then harder to eradicate. I mean, this is just one more reason why we can't ignore women when we design HIV cure types.
So if they're intact by sequencing, but does that mean if they're in silent genomic regions, does that mean that they're less likely to reactivate?
So yes, exactly. So there are two independent things. If they are intact, it means that potentially they are reactive, like they are transcriptionally
capable
of being reactivating. But if they hide into these repressive genes, it's much harder to reactivate them. And so it most likely it has to do with immune pressures, which are stronger in women. And so over time, women just select for viruses that are hiding in these graveyards. I mean, I think this is really interesting. And then this paper was also very recent from UCSF, Peter Hunt and Rebecca Edelman, and they were looking at the immune response, comparing women and men before and after menopause. So basically, they had almost 1,000 women, 1,000 people, and they are Phoenix participants. Most of them are men, obviously, because Phoenix is mostly US based. They got really lucky that somehow the median age was 47, which was perfect for a menopausal study. And they looked at an Olink platform, which is a proteomic platform with a lot of inflammatory proteins. So here you can see they looked at the anti-Mullerian hormone, which is a hormone that is most people use to try to define menopause, and they can nicely show that women lower than 47 have high level of anti-Mullerian hormone, and after 47 it decreases. So they use this to basically make the argument that this was a good proxy of menopausal transition. And but if you look at inflammation, first of all, inflammation increases much more dramatically with age in women. So if you look at this graph, everything that happens on the left is greater increase in women. Everything on the right is greater increase in men. And you can see there is a lot more going on the women's side. And then when they separated the analysis for women below and after 47, you can see that in the below 47, it's very similar, but then almost everything happened in the older women. So there is definitively something going on in the immune system with the menopausal transition. And when looking at the interferon response, which, if you remember at the beginning, is our double-edged sword, and one of the most, the strongest antiviral responses, over time, the blue one, there is almost no change in men. And when you look at women and but lower and above 47, there was a huge increase where women above 47 have more interferon, which, as we know, is good and bad, because this can probably explain the increase in mortality. And when then they looked at the upregulation of genes that were associated with mortality in women lower and after 47, all of the red dots are an increase of genes that are associated with mortality. These are all people with HIV. I think probably that's true also for the general population, but I think there is definitely a lot going on during menopause. Almost all protein that were upregulated in older women were somehow associated with mortality and morbidity. And this is how we explain the loss of female advantage in life expectancy, which I don't think is anything new, but it's nice. Like it kind of tells the story, and it makes sense that when looking at the probability of survival in women before 47 or after 47, like we have an advantage when we're younger, and then we catch up after menopause. It's basically the same as men. So basically here are a few take-home messages. Menopause might probably reshape the HIV inflammation balance. Age profoundly modifies sex difference in inflammation. The interferon response increased dramatically in women after menopause. So it looks like menopause might unmask this pro-inflammatory phenotype. And what we need to do right now, and what we're doing as part of the R1, our longitudinal study, where we have well-characterized women pre, peri, post-menopause, where we can and maybe do some single-cell RNA-seq and really look at, and I can already say, I didn't show the data here because they are unpublished, but we see something very similar going on.
So we've done well enrolling 54-24, the HOT study.
I know,
and we just did 2 physical exams for women exiting the study, and they were in love with their hormone therapy.
Okay.
Well, it was blinded, so they don't know. But they I think we're both planning to get a prescription from their providers.
It's amazing.
Really
and for those of you that don't know, right, like I was really excited about this ACTG trial where finally we're studying hormone replacement therapy in aging women. And it looks like they're doing amazing. And I was really surprised how well UCSD was doing because, I was told my entire life that UCSD cannot enroll women.
And it's up enrolled by, I think, a good chunk.
Yeah.
Shout out to Dawn. Shout out to Dawn. Yeah.
Who? John?
Dawn. She's
She's
doing amazing.
Okay. Yeah. Okay.
Yeah.
Oh, that's your person. Yeah. So, I mean, it's possible, right? Like there is more and more evidence that, for the longest time, we have said it's impossible, but then if you really want to do it, you can do it. Okay, so and here I want to show you a little bit of data about women remaining underrepresented in HIV research. These are like data from 2019, and I was looking, I couldn't find anything similar that was more recent, and maybe it's time to check again. My earliest slide had data from 2015, and honestly, between 15 and 19, there was almost no change. So basically, like this is like a landscape study where they looked at all published HIV cure trial that they could find, and they looked at which one reported sex of the participants, and then what they found is that 17% of participants were women. The median was 11%, which is exactly the same as the older 2015 study. And I can almost bet it probably didn't change up until today. In any study with antiretroviral, where they were doing treatment interruption, like 89% of people were men. What was a little bit upsetting to me is that out of 128 publications, only 45 even reported the sex of the participant, which is insane. And 18 of those 44, which is almost half, well, 40% enrolled only men. And only 2 had sex-specific eligibility criteria. And 16 enrolled only men without an obvious reason to only enroll men. So, I mean, there is a long way to go. But the problem really starts before the clinical trial. And primary cells, like the vast majority of in vitro study, even when they use primary cells, people don't even know if they're using cells from men or women. I think more, like some people right now start caring, but very often you just buy leukopacks from the blood bank that you don't know who donated this, right? And if you start creating a sample size where you mix men and women, you might be, masking some effects just because they are not the same. And similar with animal models, historically, there are a lot of animal models that rely on men because women are a bit messier, also the female animals, and also, for example, for non-human primates, very often, because they're very expensive, they like to keep the female as breeding stock, and so they do focus on men. And I think that every time we don't do a sex-specific analysis, even in preclinical trial, we miss an opportunity to learn what is going on. And early stage clinical trials create an additional barrier for women, because obviously there is no, unknown reproductive risk, and contraception requirement then become crazy, and people are requested to have like 3 different contraception requirements, and they are enforced independently if the women have sex or not. Like I know women that never have sex with a man in their life, and they are required to do contraception. So basically, there's the safety first exclusion, and so this is how we end up with a lot of early phase clinical trial with very few women. And then we self-perpetuate this gap. And obviously, enrollment requires designing study with women in mind. And I think everyone knows what that means. And I see it looks like, like at UCSD, we are doing a much better job, but women have different needs. They need different outreach. There are different stigma, privacy concern. It's a little bit harder, right? If we have to keep in mind the menstrual cycle when we do the blood draws. Some sex-specific analysis can be complicated in women. Like, let's think about genital secretion collection. It's much easier in men, of course. But the bottom line really is that when we exclude the women, we don't do a favor, even if it's a little bit more complicated and it needs more money. And this is not only at the site level, but at the sponsor level, of course, that we kind of need to design the study with women in mind. And why does this matter? I think this is a perfect example. Like we all know Karina Dubé, who was a faculty here, and then she moved to Philadelphia. But she is a social behavioral researcher, and she spent the last years like really terrifically advocating to about the treatment interruption trial in South Africa, because the ACTG network was starting to do some of the clinical trial in South Africa, and they were also doing treatment interruption, but the population is completely different, right? And we had a lot of experience doing treatment interruption in the US with men, and now they were going and doing a first trial with treatment interruption, enrolling basically only Black young women. And Karine was the fierce advocate, and she did so much groundwork to just show that it's completely different. A lot of these women, they are afraid, they are coming from abusive relationship where they experience a lot of intimate partner violence, and for them, even disclosing their status puts them at risk. And so if you ask them to interrupt antiretroviral therapy without disclosing, it makes it so much, there is much more anxiety involved and much more stigma. And she created really single-handed almost, created a whole, she published, I don't know how many. I was actually looking for a study that I wanted to show, but there was so many I couldn't pick. But I think that this is a perfect example about why you can't just do what works here and move it to Africa. And if you don't have like a champion like Karine that really put down her feet, I don't know how much we would have done, and maybe something could have gone wrong. So how do we move forward? So first of all, I think it's really important we design the sex differences from the start. Also preclinical, clinical research at every stage when you design a clinical trial. And I think ACTG is getting better at it. They have questionnaire and check points where they force team to think about, for example, for my trial, we do like at least 25% women. I mean, we can't be too ambitious in the US because obviously there are not so many women, but let's try at least to match the epidemics and not, set the bar too low. Be very intentional in enrolling women. So we know that if we the outreach that works for men, very often, if we don't change anything, we will end up only with men. Power the study to examine for sex and not just only adjust for sex, really do a real stratified analysis. Report sex disaggregated data, even if it's 0. I mean, shame on you, but I think it's important to say that that's when I review a paper and I see there is basically no women, I force them to change the title instead of like in people with HIV, the title should be in men with HIV, right? And then engage women as a research partner from the very beginning, from design to implementation. And we published quite a bit of very nice studies. In 2022, we have this community call to action to prioritize inclusion and enrollment of women. There are many more details, tips about how to try to enroll women. It's a complicated issue. I'm not saying it's easy and we can solve it overnight. Karine and myself, we have this other survey, qualitative interview-based study in the US. We have this beautiful, beautiful opinion piece about vulnerability that I really want everyone to read, because we wrote it. Jeff Taylor was the one who kind of really inspired us to write it, about how we can reframe the concept of vulnerability in HIV research. And we didn't have so much women in mind. It was more for end-of-life research, but really women are the same, right? Like, let's not use vulnerability in a patronizing way to exclude people, but let's use it as an opportunity to sit down with community leader and see how can we make the research acceptable and inclusive. And then obviously again, the Lizzie paper, which now everyone will have to go and download because it's really good. And then, I mean, obviously here I did focus mostly or almost exclusively on cisgender women, but it's really important to note that when we talk about women, we really should talk about all genders. And we all know that gender identity has nothing to do with sex assigned at birth, and exogenous hormones can influence, as not most likely influence HIV persistent and immune biology. Transgender women, non-binary people, transgender men, like everyone really should be studied one-on-one if we want to have a cure that really encompass everyone. And like, shout out again to Lizzie, because she got her K23 notice of award less than a month ago, and that's a big part. And so if you want to hear more about transgender, I'm sure she will be happy to comment.
Although you won't find that word in the in the application.
No, you word, but she has really been shaping her career around it, and she's doing a fantastic job despite all the limitation. And so the missing piece, just to finish, women remain underrepresented in HIV cure research. I think right now we can't ignore the facts that sex and sex hormones shape HIV persistence and the immune response. Like the menopausal transition right now has been studied more and more, but it looks like it's really a crucial piece in women's life. And I mean, so is, puberty, and so are pregnancies and lactation, right? There is a lot more that we need to study, and who we study determines what we can discover. And here's just to finish, we cannot all succeed when health laws are holding back. This is one of my favorite quotes. And so there is a lot of people that I want to acknowledge, but I also want to really take a minute to recognize Lorraine Willenberg, who was one of very few exceptional elite controllers that was known for eliminating the HIV reservoir without any intervention, only with her own immune system. And she was known as the San Francisco patient at the beginning, and she passed in April. She was an amazing advocate, and she donated her body to Last Gift. And right now we are studying all the tissues, and she really inspired a lot of the work and advocacy around involving more women. She was really vocal about it. And this is when we went to sign the informed consent in December. I went with Andy and Susan to visit her, and yeah, thank you.
Well, thank you wonderful talk. We've got quite a few claps and loves. There are comments that I'll read from Maya Whitaker from the NIAAA. What a great slide. I think you were showing the slide about the sex difference in the studies between men and women.
I can share the slides, by the way. Yes.
Okay, great. And there was other comments, male biased animal model studies, sex and the age of the animals study do affect disease phenotypes. Thinking back to COVID and the vaccine trials with sex difference exploration, we learned about vaccines, temporary changes to menstruation, sex differences, exploration is so important. So agree, sex differences from the start. I think you were talking about that, human and animal studies necessary. Green says, Sarah, wonderful talk. I wonder if there are any data on sex differences in the last gift.
That's a good question. So we are not doing so I'm not saying we are doing everything we can to enroll women. It's hard. In the last gift, and I think we have a handful, and we are studying them. And I don't think we have been powered so far to do a proper sex difference.
Sarah, why do you think it's hard to enroll with
Cheryl, do you want? Cheryl and Steph? Why is it?
I think it's hard identifying them more than actually pitching the study to them.
But different than it is like in anything we do where we're trying to enroll women in research, like does it feel added? Something is added to that?
You're saying you're just not getting the referrals and
Yeah, I mean, that's a little bit of it, I think. I wouldn't say we have like a higher rate of like decliners or anything like that amongst women. I think we just don't have as many.
Once we find them, we can get them on study.
And once we find them, we go above and beyond. Like we always say, if there is
Why go above
women,
and beyond for everybody?
But you know, like we
Just
gotta
for
represent someone in Orange County, and so we've been driving up to Orange County to see them because we just don't get that many women referrals, so.
Isn't it like the controls are more, it's been easier to.
It's been easier to enroll control women than women with HIV.
So that's what I'm trying to understand.
But I
what
think
you
that was.
Is there.
But also the controls, they quite honestly have sort of fallen into our lap.
Yeah.
Like via, they're one of the caseworkers across the street. Like we just had a small talk because of Laura Bamford, and then he referred somebody for a control. And then another one was a friend of a friend. So I can say that,
No, I'm winning the conversation. I have another question.
Oh, no, let me just, we definitely try to do the best we can here in San Diego with the understanding of the limitation, but what we're trying to do is kind of expand the last gift. We have been in talk with a site in Sao Paulo. They have more women and they are really excited to get a rapid autopsy. And we have been approached by NIMH if we wanted to try to be like a training site for other sites. And I was hoping that maybe if we can set up like a rapid autopsy in the South where there is more women, I think that maybe that, also it's not either or, I think both. And maybe we should sit down and just talk about what we can do better, but also I think expanding to sites where more women are available.
Yeah, well,
Maybe do you have any women? I mean, are you our top
I do
prescriber?
have some. I did have one that recently passed, but she wasn't interested. I'd spoken with her about it, but I'll definitely keep that more in mind.
Well, and I will say at the beginning of the study, we were focused on, can we recruit people? Can we get enough folks on? And so it was easier to recruit men just because there were more of them. So we did that. But I would say in the last 2 to 3 years, we really focused on getting more women involved, getting more minorities involved in general, so that we can have that diverse population within the last gift. So I think part of it was kind of
right?
we've done, yeah.
And I think y'all are wonderful, but I think we're seeing what happens for all studies, right?
Yeah. No, I
Start
know.
with the low hanging fruit, but
Yeah.
that doesn't,
Yeah.
that's not always a great
Yeah.
approach. It's what we have to do.
And-
Yeah.
But I just challenge you to think about maybe why you're having trouble with enrolling, because, right, I mean, it should be 10% here, like we do for any.
Well, and I would say we have
Usually we have
that.
10%.
Yeah.
It's just not
Yeah.
a high enrolling study, right? We have,
Yeah.
right now we did 42 autopsy and maybe 5 are women, right?
Okay.
So we were not doing terribly.
Yeah.
It's only that, because we only have 40 autopsy or
44.
you can't do a lot, we're still underpowered. And also because we started later enrolling women consistently, I also think we are behind with generating data, going back to Karin's question.
So we are getting a message from Ashley Welder. I have to log off, but if we have female patients who might be interested in being a part of research, what information can we give them to connect to you? I work at the VA. Can you email me? And so there's an email. Ashley Wilder. I have a handful of females who I think would be interested.
In Lafayette?
Probably.
Okay.
So make
Take-
sure you
copy
keep
paste
that.
the
one.
email
But
so we
we're
can-
gonna, I think Erin had a question and I know Gigi had her hand up, but let's go with Erin first. And I'm gonna let Jill finish the moderation. This, all right, Erin.
Thank you. The comment about control has me thinking, I appreciate more this morning the effect of estrogen on HIV viral life cycle, but I'm thinking about the inverse and the effect of HIV on estrogen regulation. Are there any studies that are looking at, I'm thinking hypogonadism in my cis male patients, but are they seeing or looking at similar estrogen differences in somebody living with HIV? Perimenopausal changes occurring earlier or later in somebody who's living with HIV infection?
Well, that's a very good question. I can imagine that probably there are probably some bidirectional, nothing is easy. I don't know it from the top of my mind. I had to look into it, but I can imagine that most likely everything is bidirectional, right, in biology. And right now we're studying the effect of estrogen on the virus, but you're right? Men with HIV are more likely to have hypogonadism. So there must be some effect there, which I don't know now the mechanism not from the top of my, do you know?
No.
I don't know if
anyone
knows.
I don't think it's,
Recently couldn't find it.
but at least we know that it's there, and it would be nice to know the same for estrogen, if there is.
I just want to point out one really great change that I've seen in the last 10 years. Remember there was the dolutegravir neural tube defect issue, and there was the thought of withdrawing dolutegravir from women worldwide, and then women around the world said, "Stop." We want to be part of this discussion, and we want the best antiretroviral, and then it turned out not to be correct. But then just the way that the cabotegravir studies and the lenacapavir studies for PrEP just immediately included large scale studies with women who became pregnant, lots of pregnancy data to show that it was safe. I mean, I couldn't imagine that 20 years ago.
Yeah. In fact, one of the PrEPs is still only for men, right?
It's very different for the Descovy.
Yeah.
The Descovy where they didn't really, and the one where they said in cisgender and transgender women, and then you looked and it was 1% transgender women. It's
Right.
you can't
Well,
put that in the title.
I mean, I don't think it's ever gonna get updated, but we know that Descovy works for cisgender women
Yeah.
from the Purpose One trial. But I don't know if there'll ever be an indication. So that's always gonna be, and sorry, I know there are some Gilead folks here. I feel like that is a little bit of a strike
Well,
against
they've
them. That
redeemed with
being said,
lenacapavir.
exactly, redemption.
Yeah.
Yeah.
But I mean, I think it's also that
Yes,
things
it's
just
just things
changed, like
change,
the
right.
perception and the need to have more inclusive enrollment.
Right,
I think it has evolved a lot over the last 10 years.
right.
Yeah, and for 5386, so we were the top enroller at UCSD for that very complicated cure study. And we had such trouble enrolling the study. And then it was well into it, we're like, oh my God, we're not enrolling any women. I mean, it was just really, and we had very specific enrollment targets. And so there was a lot of reflection about why that happened. And once we started pressing the sites to really focus on enrolling women, we did get an uptick at the end. So I think we got around 10 or 15% total. But yeah, that was a missed opportunity along the way.
This is a very good example about how you don't enroll women by accident. It needs to be intentional.
Yeah.
In the US.
Yeah.
Yes.
And then there were lots of things that biased against women. So we had leukapheresis, and so we had several women that didn't have the vein size to do leukapheresis, for example.
Yeah, that's a good point that I didn't think about, but someone recently brought it up that every time we require a leukapheresis, we already kind of make it harder to enroll women.
Yeah.
I 100% agree.
Yeah.
Because we have that problem here.
So you see how it tickles in every direction, and half of the time you don't even think about it. So we always want leukapheresis because then you get a lot of cells and you can do a lot of studies. But then I didn't know about it until like maybe 2 years ago.
Yeah, I mean, if we had to do over again, we would have had the optional large volume blood draw, which is no picnic either. That takes forever.
Yeah.
If people couldn't do leukapheresis.
Gigi had her hand up. But Gigi, are you there?
Yeah, hi, Jill. Sarah, great talk. Thanks so much. I have just a couple other thoughts about enrolling women. One, Dawn, I want to applaud Dawn. She has been amazing about reaching out to me. She has been a pleasure to work with. And you guys are always welcome at our FEMON meetings or to reach out. But my other thoughts are the difficulties. I still feel like there's more stigma in women. I still have more female patients who have not disclosed their status to anyone. And now with ICE issues and the Haitians losing their TPS, we have more and more patients where who are afraid to come to appointments, come to get their blood drawn. So the challenges to reach out to women, I think, are worsening right now. But anyway, I'm thrilled to hear, that this is a growing area of research and happy to help in any way I can.
Yeah. And we will reach out. Sharon will reach out and stuff.
She's given us a lot of referrals.
Okay.
Yeah,
thank
she's
you.
another really good referral.
Thank you. Thank you, everyone.